Hi guys, my name is William Zhang and our group will be presenting the paper 3D Printing of Hybrid Biomaterials for Bone Tissue Engineering. I will be presenting the background and aim for this experiment. So first I'll provide a little bit of background information on bone. So bone is an inorganic mineral with an organic template. So because of this, a biomaterial that mimics bone should have an inorganic scaffold that provides the mechanical stability to the implant and a polymer that provides the scaffold with functional properties. These functional properties can be things like biocompatibility within the body and activity that induces the proliferation of cells and promotes regeneration. But the thing is, these hybrid materials are very expensive, and also implants need to be prepared for individualized applications. The solution to this is 3D printing technology, and 3D printing technology can give us a lot of control when it comes to fabricating these bone scaffolds. Right now, the problem with 3D printing scaffolds is that no bio-ink to print the scaffolds has been developed. Right now, researchers are trying to enrich bioinks with organic growth factors that help speed up cell growth and differentiation. A potential growth factor is polyphosphate, or poly-P, which acts as a growth-promoting polymer found in almost all animal cells, but it's especially in the platelets and serum. If bone cells are exposed to poly-P, they have increased ATP synthesis as well as fast proliferation and differentiation, but the poly-P itself is not strong enough to match the mechanical properties of bone. The aim of this experiment is to develop a bioprintable scaffold that has stronger mechanical properties than just poly-P. To do this, a polycaprolactone, or PCl, with poly-P scaffold was used. PCl needs modifications to improve its biocompatibility as it's not especially biocompatible with the body. And this is done by forming a hybrid with poly-P, which is in fact very biocompatible. So essentially we are combining the strength of PCl with the biocompatibility of poly-P. So this experiment investigates this as a hybrid material that can form bioprintable scaffolds.
Hello everyone, my name is Gary Lin and I'll be walking you through the materials and methods used in this study. First we begin with the formulation of the PCl calcium polyphosphate microparticles composite. Ethanol dissolved calcium chloride dihydrate was added dropwise to sodium polyphosphate dissolved in water. After being stirred for 5 hours, the calcium polyphosphate microparticles formed in the solution were then filtered out, washed with ethanol, and then dried. Then, pre-purchased powdered PCl and these microparticles were mixed together and sieved multiple times to obtain a homogeneous mixture. To create the scaffolds, a small amount of the composite material was put into a 3D bioplotter printing cartridge and allowed to melt in the head at 100 degrees Celsius. After melting, more aliquots of composite was put into the cartridge in melting cycles until the cartridge was full. The strand thickness, spacing between strands, layer thickness, and strand orientation was controlled through bioplotter software as the scaffold printed. Of particular note is the rotation of each layer printed at an angle of 45 degrees. The mineralization, defined as the deposition of inorganic minerals on an organic matrix of the composite and the control PCl, was tested through the submersion in simulated body fluid, which contains an ionic composition similar to that of human blood plasma. The scaffolds were incubated inside of sealed tubes containing these fluids over a 4-week period at 37 degrees Celsius. They were then removed, rinsed, freeze-dried, and then analyzed microscopically and spectroscopically. Nano-indentation techniques were used to test local properties of the scaffolds. In this setup, a diamond indenter was inserted at a constant loading rate before being held at a maximum load for 30 seconds, 1 micrometer into the sample while constantly collecting data. Over at least 20 independent measurements were then performed at different sites to obtain Martin's hardness and Young's modulus calculations. Then, to obtain the overall mechanical properties of the scaffold, a force testing system was used. Due to the scaffolds used in bone and the natural and isotropic nature of bone, the measurements were conducted longitudinally to the cylindrical scaffolds. In the compressive loading setup, a 95 Newton force was applied for a minute on the sample before being unloaded for 5 minutes. This was repeated 5 times to calculate the bulk Young's modulus. Creep recovery was also tested, in which 25 newtons were applied for 30 minutes before allowing a recovery period for 10 minutes. To analyze the interfacing behavior between the scaffold and human cells, cell suspensions composed of primary human osteogenic sarcoma cells were added to the sterile scaffolds in 24-well cell culture plates and allowed to incubate for 3 days. After incubation, the cells were stained and examined using digital light and scanning electron microscopy. Finally, tests were performed to determine the steady-state expression of the gene encoding the STF1 chemokine protein in primary human osteogenic sarcoma cells. The cells were incubated in wells with and without scaffolds over a 3-day and 7-day incubation period. After these time intervals, the cells were harvested and their RNA was extracted. Then, a quantitative reverse transcription polymerase chain reaction was conducted to determine the steady-state expression of these genes.
And moving on from methods, I'm Desmond Young, and I'll be presenting the results section. We'll first look at the Fourier transform infrared spectroscopy plot. These figures help to display chemical composition and bond types of different compounds. For our purposes, it helps to confirm the fact that the PCl calcium poly-P scaffold contains elements of the PCl as well as the calcium poly-P scaffold. Some important similarities to take note of are shown on the slide. Digital light microscopy provides high-resolution images of the scaffold structure. Compared to the PCl scaffold seen in panels A and B, the PCl calcium poly-P scaffold shown in panels C and D shows a smoother surface and a rougher texture. Both local and bulk mechanical properties were tested for the PCl and PCl calcium poly-P scaffolds. Using a nanotest nanoindenter system, the researchers found a 119.2% increase in Young's modulus, determined by the slope of the displacement load diagram, and an increase of 225.8% in Martin's hardness of the PCl calcium poly-P scaffold compared to the PCl scaffold. For bulk properties, both scaffolds exhibited similar measurements. The PCl and PCl calcium poly-P scaffolds had compressive moduli near 4.0 megapascals, again determined by the slope of the stress-strain graph, and a maximum strain of around 30% when subjected to a creep. In order to assess biocompatibility, the scaffolds were subjected to in vitro growth studies of SAOS-2 cells. Controls for both scaffolds showed no growth during the 3-day incubation period. For experimental scaffolds, density and cell viability, determined using calcine AM dye, DRAQ5 dye, and fluorescence spectroscopy, was significantly greater for the PCl calcium poly-P scaffold, panels E and G, than for the PCl scaffold, panels F and H. Scanning electron microscopy, or SEM, was used for closer inspection of cell density and morphology. The seeded scaffolds were assessed after 3 days of cell incubation. No cells were discernible for the PCl scaffold, shown in panels A through C. In contrast, the PCl calcium poly-P scaffold showed abundant cell growth as seen in panels D through F. Living cells are highlighted with a black C for the PCl calcium poly-P scaffold. After depositing PCl and PCl calcium poly-P scaffolds into simulated body fluid for 4 weeks, the researchers used energy dispersive X-ray analysis, SEM, and FT-IR spectroscopy analysis to determine the mineral growth of HA on the scaffolds. Hydroxyapatite, or HA, is a major constituent of hard tissue, such as bone and teeth, and is therefore desirable for implant coating. Previous research has shown a 1.6 calcium to phosphate ratio is needed for HA development. From panels C and F in the figure on the left, it can be determined PCl scaffolds contain mineral deposits with a 0.6 calcium to phosphate ratio. PCl calcium poly-P scaffolds, on the other hand, exhibited a ratio of 1.1, closer to the HA formation requirement. On the right is a FTIR spectroscopy graph indicating these results, with the HA characteristic signals marked with white on black. Finally, the expression of the SDF1 alpha gene, encoding a chemokine protein involved in migration, was determined as a function of scaffold exposure. PCl scaffold showed no change in gene expression from the control for the 3 and 7-day incubation period. In contrast, the PCl calcium poly-P scaffold exhibited upregulation of the gene at values of 3.8 times for the 3-day incubation and 6.8 times for a 7-day incubation period. Thank you, and on to Zach for discussion.
I'm Zach Spalding, and I'm going to be presenting on the discussion from our research article. From Desmond's coverage of the results section, we've really been able to appreciate the unique and ideal properties that this tissue scaffold holds. The PCl-Ca poly-P material tested throughout this experiment allowed for massive improvements to the mechanical properties of the scaffold in comparison with solely the PCl scaffolds, as shown in this figure here. Farther, this was accomplished without sacrificing selectivity on the scaffold. In the presence of the SAOS2 cells, the PCl-Ca poly-P mixture was able to show significant improvements to cell cultivation when again compared to solely the PCl scaffold. With such promising results, there are many opportunities for the study to be expanded into broader and more diverse testing environments. Only so much testing can be done on the mechanical properties of the material, but monitoring cell growth in different exposures could be very interesting. As mentioned before, this experiment monitored cell activity on the scaffold in the presence of SaOS2 cells. It would be interesting to see if other identified cell types in the site of scaffold placement would elicit different responses over time, and if this requires any properties of the material to be tweaked. The study could also be taken in the direction of animal testing for further results. Many tissue engineering studies work with mice or rats to observe experimental results in larger living subjects, and this could give the researchers very important insights that were missing with the solely cell exposure. Further down this path, and on a longer time scale, if the results continue to show ideal tissue growth with little problems, clinical testing could be a route to allow this material to assist struggling human patients if successful. Farther steps could also be taken in modifications to the researchers' existing experiments. This figure here shows an observation of gene expression and selectivity with the scaffold over an incubation period of 7 days. But how do these results look on a longer time scale? The researchers mentioned that PCL is biodegradable and has a degradation timeframe of greater than 24 months, so the observation of the scaffold's interactions with cells over a period of multiple weeks or months could show some results about the scaffold's long-term degradation profile. In a long-term study, we also want to consider biocompatibility and undesired cell responses. The PCL scaffold is found to remain non-toxic during degradation, but the monitoring of cell toxicity would be interesting to see on the longer time scale, especially since the scaffold is not solely PCL. On a big picture scale, the researchers have shown that this PCL-CA poly-P mixture for those tissue scaffolds is more than just another scaffold biomaterial with slight modifications. The researchers have been able to innovate the production of the scaffolds in such a way that they can easily produce a very ideal biomaterial through 3D bioprinting, which is very impressive. As the researchers so very well put it in the first line of their article, this article describes the creation of a morphogenetically active bioink, which is much more than an ordinary biomaterial. We think this article has shown a very promising solution to the issue of bone tissue regeneration through scaffold-mediated cell growth and has a high translational potential in a medical context. Though the 3D printing helps in physically producing the scaffold, we believe this may limit the scaffold in its shape and 3D configuration. The printing system can make circular configurations, but for situations requiring more complex shaped scaffolds, we believe that 3D printing would not be ideal for scaffold construction. Other groups of researchers have created similar 3D printed bioactive scaffolds. In one study, a scaffold was printed that had innate photoluminescent properties due to the materials used in its construction, which we think would be an improvement over the previously discussed scaffold. It exhibits successful mechanical and cell growth properties while allowing easier real-time bioimaging in vivo due to the photoluminescence. Overall, this field has shown itself to be a source of intrigue and future advancement in the biomaterials field. Thank you for listening.